Good day to all of you. So today I would like to explain and to share knowledge with you how to do the purification and also the characterizations of the one enzyme, amylase. Okay, first of all, do you know what is amylase actually? Do you know? Okay, actually amylase is an enzyme. You can see the structure behind beside this okay, actually is enzyme. It's a protein structure where in the pictures you can see the catalytic site and also the active sites. So actually enzyme is a protein, but not all the proteins is enzyme. You need to remember this is really important. Okay, next, when we talk about the amylase, when we talk about the enzyme, when we want to purify the enzyme, we need to know this enzyme actually is an extracellular, intracellular, or a membrane-bound enzyme. But in this term, amylase is one of the extracellular enzyme. Okay, there are two types of the enzyme amylase. First type of the enzyme amylase is alpha amylase. Alpha amylase, we know that alpha amylase is used to break the alpha-1,4-glycosidic bonds, and it can hence in production of the dextrin and some of the oligosaccharide. However, the beta amylase is only can produce some of the dextrin, but also the same is to break the alpha-1,4-glycosidic bonds. Okay, when we talk about the functions of this enzyme, amylase is used in some of the food factory, and also some of the food factory they produce the high fructose corn syrup, and also they use in the pharmaceutical. And do you know there are amylase in your mouth also, as well as a digestive enzyme. So this is the hi. In this second part, I will tell you about what actually can produce this alpha amylase. Okay, in producing this alpha amylase, we got three main sources. First is the plants, second one is the animals, and the third one is the microbiome. Okay, the first one, plants, as the journals, they stated that we can also get the amylase from the wet. Also right, but the second one is animals. Some of the animals can produce amylase. Actually, we also can produce amylase from our saliva. Okay, the third one is a microbial sources of the amylase. But in this case, we take the Bacillus subtilis. Okay, why we actually use the Bacillus subtilis? Bacillus subtilis actually has some characteristic that make it suitable for the factory to use this Bacillus subtilis as the producer for the amylase. First is the Bacillus subtilis is heat resistant, and the second one, it can produce a lot of endosperm, and the third one is it can produce a lot of amylase. So this is one of the reasons why we use the Bacillus subtilis. So in this, after this, you will see the process of the purifications method and also the characterizations of the Bacillus subtilis and on producing the alpha amylase. So stay tuned.
To obtain alpha amylase, Bacillus subtilis is cultured first. First, inoculation takes place whereby one loopful of the strain is inoculated into a nutrient broth containing distilled water, beef extract, yeast extract, peptone, and sodium chloride. Incubation is next, and lasts for 24 hours in a shaker for proper growth and development of Bacillus subtilis. Next, inoculum transfer is done aseptically into 100 ml of production medium. The medium contains peptone, yeast extract, monopotassium phosphate, magnesium sulfate, calcium carbonate, sodium chloride, starch, and distilled water. This combination provides carbon and nitrogen sources and also a pH of 6.5 to 7.0 for optimum growth. The addition of starch acts as a stimulant for amylase production. After inoculum transfer, the medium is incubated in a shaker for 72 hours to obtain a cell suspension from fermentation. Purification is done to recover alpha amylase effectively from cell suspension resulted from fermentation in minimal steps. The aim of purification is to increase the specific enzyme activity of alpha amylase and decrease the total protein. Purification steps. The first step of purification is to concentrate the cell suspension obtained from the previous steps under nitrogen stream by using ultrafiltration with a membrane of 100,000 relative molecular weight cutoff. As alpha amylase is an extracellular enzyme which would be present in the supernatant, the filtrate is collected while the retinate, which consists of microbial cells and are unable to pass through the membrane, is discarded. Filtrate collected is reconcentrated with membrane of 30,000 relative molecular weight cutoff. As alpha amylase has a molecular weight larger than 30,000, it will be retained as retinate and the filtrate containing small contaminants is discarded. Next, the retinate is subjected to protein precipitation where solid ammonium sulfate is added slowly with constant stirring at 4 degrees Celsius to the enzyme to give a final concentration of 40% saturation. It is then kept for 18 hours at 4 degrees Celsius. By centrifugation at 35,000 G for 15 minutes at 0 degrees Celsius, precipitated proteins are separated from the supernatant containing ammonium sulfate salt. After dissolving the precipitated proteins in minimum volume of 0.1 molar phosphate buffer with pH 7.5, dialysis is conducted against same buffer by using molecular porous membrane tubing for 18 hours at 4 degrees Celsius. Next, gel filtration chromatography is carried out whereby a Cephalose Cl6b column is pre-equilibrated with 0.1 molar phosphate buffer with a pH 7.5 before applying dialyzed alpha amylase sample onto it. The column is washed with 500 ml of equilibration buffer and the bound protein eluted with the same buffer. Fractions of 2.0 ml each are collected at a flow rate of 0.5 ml per minute. Next, the fractions collected are assayed for enzyme activity. The active fractions which show extracellular alpha amylase activity is pulled and concentrated by lyophilization. At the same time, pooled active fractions collected is applied to high-performance liquid chromatography for purity check analysis by HPLC using a carbon-18 reverse phase HPLC column. The chromatogram of purified enzyme is then compared with standard alpha amylase. If similar retention time is obtained and there is only a major peak, it can be confirmed that alpha amylase has been purified to homogeneity. By using sodium disulfide polyacrylamide gel electrophoresis, the molecular weight of alpha amylase is determined by comparing its electrophoretic mobility with that of standard proteins having known molecular weights. Only one single protein band should be shown in SES page profile of purified enzyme. Previous study has shown molecular weight of extracellular alpha amylase from Bacillus subtilis KIBGEHAS to be 56 kilodalton.
The alpha amylases obtained from Bacillus subtilis were purified by a series of treatments. After purification, it showed a single band on SDS page. The optimum temperature of alpha amylase was found to be at 45 degrees Celsius, while the optimum pH was found to be at 6. Enzymes showed Michaelis-type kinetics when hydrolyzing soluble starch. Kinetic parameters were calculated as 1.08 mg per milliliter for the Michaelis constant and 100 U per milliliter for Vmax. The effect of metal ions on alpha amylase activity was measured in the presence of various metal ions at a concentration of 1 millimolar and 5 millimolars. The 1 millimolar concentration was more effective than the 5 millimolars. Activities of alpha amylase were stimulated in the presence of calcium, barium, magnesium, lithium, and manganese ions. On the other hand, a strong inhibitory effect was observed in the presence of copper, mercury, and silver ions. The amylase did not require any specific ion for catalytic activity. Molecular weight of purified alpha amylase was found to be approximately 56 kilodalton. The 20 residues of the amino terminal sequence were determined to be V, N, G, T, L, M, Q, Y, F, E, W, Y, T, P, N, D, G, Q, H, W.